We can't answer these questions. Neuroscientist Kenneth Kosick on whether lab-grown brains will achieve consciousness. Well, we know that, you'll see a picture in a little while, brain organoid biocomputers. They use human brain cells to help these computers work faster and better. And the way that they've coaxed these little brain cells to work properly and faster is by actually putting them into pain mode if they don't work well enough or fast enough. And they found that these little brain cells, these brain organoids that are used for these biocomputers, do feel pain. They do feel pain. So this neuroscientist says, well, we don't know if they will achieve consciousness. Well, if they feel pain, of course they understand what pain is, which means they have emotions. So much is still unknown about consciousness, never mind whether brain organoids will achieve it, explains a leading neuroscientist. Brain organoids are 3D lab-grown models designed to mimic the human brain. Scientists normally grow them from stem cells, coaxing them into forming a brain-like structure. In the past decade, they've become increasingly sophisticated and can now replicate multiple types of brain cells which can communicate with one another. This had led some scientists to question whether brain organoids can ever achieve consciousness. Kenneth Kosick, a neuroscientist at the University of California, Santa Barbara, recently explored that possibility in a perspective article. Live Science spoke with Kosick about how brain organoids are made, how similar they are to human brains, and why he believes that brain organoid consciousness is not likely anytime soon. EC asks, what are brain organoids and how do scientists make them? And Kenneth says, a brain organoid is made from stem cells. You can take any person and convert their, say, skin, fibroblasts into stem cells, and then differentiate them into neurons. It's what stem cells are all about. Stem cells are called pluripotent because they can make any cell in the body. We spent a fair amount of time before organoid technology came along, taking human-induced pluripotent stem cells and inducing them in a two-dimensional array to look at neuronal differentiation. So that takes us halfway there, but it only gets us as far as 2 dimensions. And then the big insight, which came from Yoshiki Sasai in Japan and Madeline Lancaster, who was to take these neurons that were beginning to differentiate cells relatively early in development and put them in a drop of what's called matrigel, a gel that can be either a liquid or solid depending on the temperature. So the cells are in this drop, and then the magic happens. Instead of growing into 2 dimensions, they start growing to 3 dimensions. It absolutely fascinates me that when biology begins to explore the 3rd dimension, a very novel biology emerges. Certainly, in 2 dimensions, these neurons that were growing could achieve a very wide diversity of cell types, but they did not achieve any kind of interesting anatomy. Once they're growing in 3 dimensions, they start to form relationships with each other, kinds of structure and anatomy that has a very loose resemblance to the brain. And I really emphasize the word loose because there are people that use a misnomer for brain organoids and call them mini brains. They're not brains at all. They are organoids, meaning like the brain. A question where we are keenly interested in, and many labs are, is that if organoids are like this brain, are like the brain, to what degree do they resemble the brain, and to what degree do they differ? And they differ from the brain a lot, so you have to be very careful about interpretations of organoids. Not everybody thinks that organoids are going to be informative for neuroscience, because what we find in an organoid may be overinterpretation. But on the other hand, it is forming a 3-dimensional structure that has some degree of lamination, formation of layers of cells within tissue. It has these rosettes in which from the center of the rosette you can progressively see cells becoming more mature as development proceeds, which is very similar to what happens in the brain, they said. EC, are there any brain organoids that actually capture the whole brain yet? KK said, there is no organoid that captures the whole brain. There are approaches that attempt to capture more of the brain than, say, just the one part that maybe we in other labs are working on. These are called assembloids. Scientists take stem cells and differentiate them down to pathways that may make a little more ventral, that's the front part of the brain, or a little more dorsal, the back part of the brain, and they put them together, they fuse them so that you get more comprehensive fusion, a wider representation, I should say, of brain anatomy. There are other ways of making organoids that are a little more indiscriminate. They're not directing the stem cell towards dorsal and ventral, they are putting them all together. That's a lot of what we do. Those were the techniques that were originated by Lancaster. And in that case, it's my opinion that when you do it that way, you get a broader representation of cell types. That's what you gain, but you sacrifice anatomical accuracy because when you make an assembloid, the anatomy is not great. But when you do it without differentiating towards dorsal and ventral, and you put it all together, the anatomy becomes even more problematic. Question, as you allude to, these organoids are similar to human brains, but there's some key philosophical differences. Can you explain? Kenneth said, so one similarity right away is that you see a lot of spiking going on. It's quite remarkable, and underlying this is the notion, which is probably what intrigues me the most, that all of this activity is spontaneous. It just arises based on the assembly of the neurons. And now we can look at the relationship of those spikes. When you do that, you can ask the question, well, if I see neuron firing, that's like likelihood that I'm seeing a neuron B firing. I'm going to look at the binary relationship among all of them, and I'm doing it with a filter that when neuron A fires, and I'm only going to look at when another neuron fires within 5 milliseconds. Why 5 milliseconds? Because that's about the time in which it takes for transmission to occur across the synapse. Synapse is the gap between two neurons. And when we do that, you can see that they form a network. You connect A and B, and you connect C and D, and then A and C, and you see that the neurons are talking to each other, and this arises spontaneously. That is one example of the way in which an organoid does something that will spontaneously resemble what happens in the brain. The way I look at an organoid, it's a vehicle that has capacity to encode experience and information if that experience were available to it, but it's not. It has no eyes, ears, nose, or mouth, nothing coming in, but the insight here is that the organoid can set up spontaneous organization of its neurons so that it has the capacity to encode information when and if it becomes available. That is just a hypothesis, Kenneth said. A question, do you think that brain organoids will ever achieve consciousness? Kenneth said, so that's where things get a little mysterious. I think that those kinds of questions are predicted on this term that people have a lot of trouble defining consciousness. You spoke about the fact that organoids have shown some capacity to encode information, but they don't have the experience to do this in the first place. What would happen if, hypothetically, a human brain organoid was transplanted into an animal? Could it then achieve consciousness? And Kenneth says, let's break that down. Before it is transplanted into an animal, some people would say the animal already had consciousness, and some people would say it does not. So right away we get into this difficulty about where in the animal kingdom does consciousness begin. So let's reframe the question. If you then took an animal which may or may not have some degree of consciousness and you transplanted a human organoid, would you confer consciousness on that animal, or would you enhance consciousness, or would you even get something that resembled human consciousness in an animal? I don't know the answer to any of those questions. We can do these hybrids now, so it's a good question, but the evaluation of consciousness now, because of all the problems as to what consciousness is is still going to be an open question, Kenneth says. Question, do we have an idea of rough timescales? Is consciousness something that could happen in the near future after, say, a certain number of years, or is it still really uncertain at this point? And Kenneth says technology is moving very fast. One place where we may begin to push the boundary is in the so-called cyborgs or organoid interfaces. That would be one direction. That could be interesting, maybe a little bit towards consciousness, but even more so towards developing the implementation of human abilities in one of these synthetic systems. So you see where they're going with this. They could be growing from stem cells little brains that can turn into bigger brains and create cyborgs from them. Question, can you think of any obvious benefits and drawbacks of these organoids being able to achieve consciousness? Kenneth, we know so little about neuropsychiatric conditions. Neuropsychiatric drugs are developed without understanding any deep physiology. All of that could be done, I think, with organoids. I think as disease models, it could be very, very useful. The dream state that I have is to develop them as computational systems, because right now, to do the kind of very extensive computations that are required for ChatGPT and many of these large language models, these take hundreds of millions of dollars to develop. They require a server farm of energy to keep them going. We're really just running out of computer power, yet the brain does a lot of this stuff on 20 watts. So a big interest for me is, can organoids, if not solve, contribute to the huge demand that we're making on the energy system by tapping into the highly efficient way in which the brain and presumably the organoid can handle information. This is by Emily Cook on Life Science. Do you understand that they use human stem cells to create little tiny brains in order to use the energy that it creates by itself? This is, I'm sorry to say, a Dr. Frankenstein scenarios going on here. Please leave your comments and thank you for your support. Finally, support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.